introduce the course faculty here. And most important guests here are Prasenjit Sen, National Coordinator of GIAN Program. Please come to the dais. And Course Coordinator for JNU of GIAN Program, Professor Tekwani. And may I request Rohini to come to the dais. So, my name is Rentala Madhubala. I'm the host of this course, GIAN Program. You must have read about the GIAN Program. I welcome our guests, Professor Mahapatra, Kanuri Rao, Dean School of Life Sciences, Professor Prasenjit Sen, Babu, Tekwani, and Professor Patnaik, and Rohini. And I have two other hosts here, Rupesh and Manoj. And I have to say that the co-hosts have been very helpful. I'm very grateful to Rohini Muthuswamy, who has been my right hand throughout this program, and very efficient. I can't, because she'll be doing the vote of thanks, but I have to thank her right in the beginning. Because of her, this program was possible. Thank you, Rohini, for being so helpful. And I welcome all the trainees here for this program, and our guest of honor, Kanuri Rao, who's heading the Drug Development Center at, he's the chairman of the translational, head of the Drug Discovery Research Center at Translation Health Science and Technology Institute at Faridabad, which is a DBT supported institute. And I'll introduce both the guests later, the course faculty also after some time. But I am really grateful to them for accepting the invitation. And I welcome all of you, all the trainees. I know a lot of you had problems registering yourself at the GIAN JNU site, and I think all those queries and how to go about it, Professor Patnaik is here, who has been working with us on the registration and other, other various details about running this GIAN program. He's been very helpful and very kind all along, and very supportive of this program. And I'm proud to say that this is the third in the series of programs which School of Life Sciences is organizing. The other two programs, and I have to thank Professor Yadav because I got all the tips on what are the do's and don'ts about this program. And it helped a lot because Professor Tripathi was the first one to organize, and then Professor Yadav, and then myself. I'm sure you'll have a wonderful time here. And I do expect you people to be here. We'll give you the timetable and the schedule of lectures today as we go on. I welcome you all for this program. And I welcome my colleagues who are here from School of Life Sciences and other faculty members. May I now request the Dean School of Life Sciences, Professor Tripathi, to give an overview of what kind of work we are doing at the School of Life Sciences.
Welcome to you all again on behalf of the School of Life Sciences. As Professor Madhubala already pointed out, this is a third program in life sciences on GIAN program, and I hope this program will continue provided MHRD continues to fund this program. Hopefully they will do it. So we are thankful to Prasenjit Sen that we are benefited by the academic input that we are getting from others, especially distinguished professors coming from abroad and teaching to our students. And Dr. Patnaik is doing a great job in coordinating this program. One thing I must say that this is a program which has run successfully in School of Life Sciences and also in other schools in JNU. And in those who have come to life sciences for the first time, those students who are coming here, then those who are already here, they know what's going on here. But those who have come from outside, they must know that, our school is an integrated school, you know, the unique school in the country, unique department in the country, where, you know, all subjects of life sciences, whether plant physiology, animal physiology, microbiology, genetics, cell biology, structural biology, you know, you come to photobiology, radiation biology, cancer biology, developmental biology, you know, advanced human genetics, all different RNA biology, you know, redox biology, you know, you name it, cell signaling, you name it, everything is taught in the school, okay? And such a school I have never seen anywhere in the country. This is the first school I am proud to say that. This school, I have not seen a school comparable to any school, any university in the country, okay? And those who have there, you know, they have different departments, you know, the school has different departments. Each department is focused on certain aspects in a botany or geology or microbiology or genetics, but not an integrated program. So our students get an integrated approach to all the broad areas of life sciences. And and all our students have done great. And, you know, we have, our students have, you know, last 2 to 3 years, you know, our students have been top in NET program, stood first in NET program. And also, our students, most of the students, 90% of the students clear NET. So I must say this, that, you know, as a teaching is concerned, we are doing a great job. And in research also, our school is quite high in the ranking in India in life sciences. With this approach, I am sure that our honored guests who are here today, Professor Konire Rao and Professor Babu Tekwani, who are here, I am sure they will know slowly what we are doing here. And of course, Professor Konire already knows, he has already part of a special committee and all that. But Professor Tekwani will know, you know, what we are doing and all that. And we'll certainly be benefited from your input, sir, Professor Tekwani, sir. And our students will also be highly benefited from your lectures. And we will encourage all our students to attend your course. We'll ask your students who have time, and, you know, they are our PhD students also, to come attend this lecture. With these words, I conclude my talk, please. Thank you very much.
Thank you, Professor Tripathi. May I now request Professor Chintamani Mahapatra, Rector JNU, to tell us something about the JNU, to all the trainees especially.
Professor Tekwani, Dr. Rao, all my distinguished colleagues here as well as in the audience, and dear students, trainees. Who doesn't know JNU these days? Many people know. I don't have to talk much about JNU, but I appreciate this kind of GIAN program so much, because every minute one individual in India dies out of tuberculosis. About 1,000 people a day and 5 lakh people a year get infected by this. In media reports, they call it a deadly terrorist. And very recently, the Institute of Science in Bangalore apparently has come up with a new drug. And this is very pleasing to the ear and satisfying to the heart and the mind. All of you know very well that doing research and then deploying or applying research is so difficult. It is a luxury in a country like India. Why? Because our companies are so used to manufacturing generic drugs, much cheaper, hardly any risk, and the duration, the gestation period is about 4 to 5 years. But when you develop a new drug, and then you have this kind of models, BioAssays and BioEfficacy, sometimes it may take about 10 to 15 years to fructify. And then, according to some assessment, about $2 billion to put it in the market. Big investment, long gestation period, and for India, it is a luxury. But then same thing can be said about any other subject, any other field. This is our School of Life Sciences, and the Dean told you about good things about life sciences. We are a country which also has something called death sciences. We don't call it, but there are people who are doing research on types of death, approaches to death, what happens after death, how to prevent death. There are people who are doing it. So in this wonderful country, with tremendous amount of difficulties and hardship, at least the GIAN program of the Government of India is benefiting all of us to have distinguished professors from abroad, from different parts of the world, coming and sharing their thoughts, their ideas, the models they have developed, and this is how we learn. In this age of technology, somebody can say that you can send an email, get an article, read in the paper, so many books are available, you can have a Skype. Will that work? That will not. If people would read books on mathematics and become mathematicians, then why do you need classrooms? So no matter how technology has developed, ultimately this human touch, some kind of imparting education front to front is very useful. In the ancient Indian text, people would say, Guru bina jnana nahi. There is no knowledge without Guru. So there is knowledge in the books, but interacting with Guru is very useful. That is why I welcome such a program. Professor Tekwani is from the United States of America. And the US may have great scientists, lots of money. The amount of money the US government spends into healthcare is about 16% of the GDP. But it is not one of the best. In India, we don't have the capitalist mode of healthcare system as in the US, or the socialistic pattern of healthcare that we have in Scandinavia. We have our own, like mixed economy, we have our mixed system. And within this mixed system, if some of you, the trainees, who want to learn more and more about such a crucial subject, it is going to help in both ways. Number one, you're all Indians. Like Professor Tekwani, someday you'll land up in a place where you'll get more opportunity, more money, more facilities to do further research for the benefit of the mankind. So there is a job prospect or more research prospect for all of you. Or even otherwise, Mother India will benefit directly or indirectly from all this research and learning. With this, I again welcome Professor Tekwani, Dr. Rao, to this good initiative of the School of Life Sciences, and thank you for your attention.
Thank you very much, Professor Mohapatra. May I now request Professor Prasenjit Sen, who is National Coordinator of GIAN Program for the universities.
Right. I'm the National Coordinator for the GIAN Program for the Central Universities.
Okay. You corrected me second time.
It's okay. Professor Tekwani, thank you for coming over to India. I think it's a great honor to have you here. This is one of the mandate of the GIAN program, and my JNU colleagues and young friends. Well, I've been asked to speak about the GIAN program, but I think I will not do that. For the simple reason is that most details of the GIAN program are there on the net. But I'm going to tell my colleagues here and you all certain details of how this GIAN program was developed, because I've been associated with this right from day one. Now there are a few things, for example, how did the national coordinator, concept of a coordinator came up during the deliberations at MHRD. And I remember the secretary that time was, I think, Mr. Satpathy, who was also a product of our university, secretary MHRD. And So there was a proposal that all the IITs would be part of the GIAN program, all the NITs or the IACRs and things like that. And there was a proposal to put the universities under the umbrella of either the IITs or the NIITs, believe me or not. So the universities did not have any independence. I fought tooth and nail for this, and so as a result, I think Professor Chakraborty, who's the national coordinator of the GIAN program at IIT Kharagpur overall is the national coordinator, said that, okay, we'll have a group coordinator for central universities and you take it up. I said, gladly. So that's how I'm the group coordinator for the central universities. So in a lot of ways, I found how MHRD actually sort of sidesteps the universities, and these programs, particularly the GIAN program and several other programs in which I have been associated, changed the perception of MHRD towards the universities. So as a result, we started in October 2014, and by the time we started with the GIAN program, all the courses, there was 13 sections identified, 13 course topics. That's physical sciences, chemical sciences, and so on and so forth. 13 topics. So 13 topics had 13 sectional committee chairmen. So all the projects go like papers, you know, like academic papers. It goes to these sectional chairmen who then evaluates it through peer review, and that's how we have Professor Tekwani and so on and so forth. So out of the 13 sectional committees, there are 4 sectional committee chairmen from JNU itself. So that has really been an achievement, I think, of JNU during all these deliberations. So I think these are details which will not be known to anybody, and I thought that I will address that. About the continuation of the program, I think it's an absolute yes, and what is most interesting and what is most important is that our young people should come for these programs. Whether they are registered or not, that's not important. I think the main purpose, the second main purpose of this GIAN program, and that's something which Professor Mahapatra just said, without a guru you cannot have a shishya, and this is meant to handshake into a future program which will be GIAN research. So now you should have Professor Tekwani, you should actually handshake with him, exchange your ideas and evolve into a research program where you can, as a student, go over to the United States to his laboratory or start research. So this is coming. So it's my request to all the young friends is that you please get into this mode. And this is the third program from School of Life Sciences. I belong to the School of Physical Sciences. School of Physical Sciences still do not have a program, so congratulations to SLS. You've taken the lead, and it's definitely a premier school in this country, and you're all part of a premier faculty. I hope you gain from the GIAN program and progress on to make this success period in your career and your life. Thank you.
Thank you very much, Professor Sen. May I now request Professor Satyabrata Patnaik, who is the coordinator for JNU GIAN program. I'm sure he'll have lots to say, tell all the trainees as to the selection process and what were the teething problems we had for this program.
Good afternoon, Professor Mahapatra, Professor Tekwani, Professor Rao, Professor Tripathi, Professor Sen, Professor Madhuwala, Rohini, and distinguished colleagues and dear friends. So this is a happy occasion for GIAN JNU. This is the 7th course running since January 2016. And today there are 3 programs running in parallel. So I would say initially there were a lot of teething problems, but it's kind of established now. Things are much better now. And the stated objective of the GIAN, as enunciated, to tap talent pool of scientists and entrepreneurs internationally, to encourage the Institute of Higher Education in India. 2, to augment countries' existing academic resources. 3, to accelerate the pace of quality reform in education. And 4, to elevate India's scientific and technological capacity to global excellence. And to this, I add the 5th component, and that is what already has been mentioned, that there's a rapid change in the mode people are learning these days. With the tap of an app, you can learn how to learn. You can learn how to play guitar in a week. or delve deep into philosophy of course in Harvard. But I think there are three indispensable components of education: a true guru, aspiring shishyas, and a dialogue. This is what GIAN program really aspires to achieve. That's the, I mean, this over projection of technology, you know, the internet and all that is sometimes lead to a lot of confusion. And the GIAN really aspires to smash this over projection and bring in leaders in their field, and students would benefit by interacting with them. That's the sole goal. GIAN, as you said, is not G-Y-A-N, it's G-I-A-N. But in Sanskrit, GIAN is a very beautiful word, and there is actually no translation of GIAN in English. Some people say it is knowledge, but GIAN is not knowledge, something more than knowledge. It's like punya. There's no English translation of the word punya. So I hope through this GIAN program, our aspiring students would really go towards true GIAN, and that's the aspiration. Thank you.
Thank you very much, Professor Patnaik. You know, I just wanted to mention the reason why this topic was chosen. As Professor Mohapatra said, this is a topic very relevant to the Indian conditions. We have major public health problems here, the parasitic diseases, or you have bacterial infections, viral infections, and several of these infections are becoming resistant to existing drugs. We read about it all the time. And we know, those who work in the field also know, that there's a lot of resistance happening to several known drugs, and there are, for some diseases, very limited repertoire of drugs are available. So hence there is a need to be aware of what is happening, what are the diseases, how do we go about drug development. It's not an easy process. Now with the genome of most of these organisms available, life has become much simpler for researchers to find out the target, identify. So we start with identification of the targets, and then probably express the target, check its properties, and then go ahead designing drugs. So it is not by one person. Then you have to check the efficacy of the drug. High throughput screening methods have to be developed. So all this combined will give you a feel, this particular course will give a feel how the resistance to existing drugs has led to the need for developing new drugs, and what is the process you go through if you have to identify new drugs. So it's and also very important part of this is developing high throughput screening methods, which is also Professor Tekwani will talk about. Now, I think the best person to introduce this drug development topic is our guest of honor, Dr. Kanuri Rao. We know him from, we consider him as part of life sciences. Now he's moved little far off from life sciences. He's the best person to talk about it, because he's heading the Drug Development Center at THSTI at Faridabad, which is a DBT cluster. Dr. Rao, just to introduce Dr. Rao, he completed his PhD degree in organic chemistry from the University of Baroda. Subsequent to this, he joined as a postdoctoral fellow in the Division of Environmental Chemistry, Johns Hopkins University, where he worked for 1 year. Dr. Rao then moved to the Molecular Biology Institute of UCLA. He returned to India in 1988 to take up a position at ICGB. Dr. Rao currently heads the immunology group, that is his dual post at ICGB, and is the founder of the Drug Discovery Research Center, DDRC, at THSTI. Over the years, his research interests have included peptide chemistry, immunobiology and B lymphocytes, infectious disease biology, and systems biology. As you will notice, he's an organic chemist by birth, but he has moved on to biology of infectious diseases. He has made outstanding contribution in the design of synthetic peptide vaccines based on the regeneration of conformational epitopes. He has also contributed significantly to the understanding of the antigen-specific B cell selection and amplification. He has been actively involved in the development of HIV diagnostics. He is the member of different scientific bodies and organizations in India and has various awards to his credit. Some of the awards he has received over the years include the most prestigious Shanti Swarup Bhatnagar Award from Government of India, National Bioscience Award, and Ranbaxy Research Award. May I now request Dr. Kanuri Rao to talk to the trainees about the drug development process.
Good afternoon, and thank you so much, Professor Madhwala, for that very generous introduction. And good afternoon to all the dignitaries on the dais. It's really a privilege to be a part of this inauguration ceremony, and particularly on a theme that over the last couple of years has come close to my heart: bioassays, bioefficacy models. And this, as you can imagine, is particularly relevant for drug development exercise. But I think the important, I mean, it's easy to get, as somebody said earlier, to carry it away by technologies and techniques and forget about the innovative components that are required. And frankly, I personally believe that India is the best place now for out-of-the-box thinking. I mean, we live out of the box, so we can certainly think out of the box. The reason I say that is if you look at the last 10, 15 years, the drug development field has become fairly grim. You don't have many new drugs on the market. Whatever comes out are Me Too or repurposed or those kinds of molecules. And that's largely for two reasons. One, as again was mentioned, the cost of bringing a drug out is maybe $1 billion or $2. So therefore, the pharmaceutical companies are a little worried about the kind of risks involved, number one. Because if you can bring that out, it has to be in what you call a blockbuster category so that they can recover their investments in a reasonable length of time. So that rules out most of the infectious diseases. So you can see there's not that much enthusiasm for certainly developing drugs against tuberculosis or malaria and so on. Maybe some of the glamorous infectious diseases, yes. But the other more critical problem, which is intellectual challenge, is over the last several decades, we're focused on the concept of one drug for one target, right? And even your guidelines, your FDA guidelines, all have revolved and evolved around that principle that if I develop a drug, it must be against a specific target, no off-target effects, that should not hit anything else, and so on. And we always forget that some of the most successful drugs we have, including something like aspirin and so on, we still don't even know what their targets are. And the problem is, as Dr. Madhubala mentioned, the genome revolution and so on has brought in a lot of new tools by which we can understand functioning of biological systems, right? These are what you call the high throughput technology tools, whether it's genome sequencing or microarrays where you look at transcript profiles, subsequent to that new proteomics technologies where you can actually look at thousands of proteins simultaneously, and what has now been evolving very rapidly over the last few years, the metabolomics. So you can actually scan from genotype to phenotype by going from genome to transcriptome to proteome to metabolome. The point when you start looking at these, you suddenly realize life is far more complex than we've ever thought it to be. We've all been taught, I'm sure most of you have been taught when you taught biochemistry, that you have a sequence of reactions and somewhere there's a rate determining step and so on. And intuitively we have been told that rate determining step is what you want to hit if you want to develop a drug. But what happens is when you start looking at these new developments, is suddenly you start realizing that there's no such thing as a discrete pathway. They're all connected with each other. They form intricate networks. And as a result, the concept of a rate determining step has disappeared, right? You have things like feed forward, feedback regulation, and so on. And it's now like a very intricate network of functions. So the components of a cell, whether you talk about proteins and so on, are all tightly linked as a network. And when they function as a group, their behavior is far more unpredictable than when you thought of them as single entities. And it's a lot like people here. If I look at any one person, if I know that person well, I can make a reasonable prediction about what that person is going to do. But put him or her in a group, then suddenly they become less predictable. And that's exactly the challenge we now face with drug development. Because the diseases we deal with today are those that persist for many years, and they're chronic diseases, right? Whether you talk about diabetes, obesity, cancers, and even infectious diseases like HIV and so on. So they're there for a very long time. So they're engaged in interactions with molecules of your cell, or perturbations of, of networks of interaction of proteins in your cell over a very, very, very long period of time. And as you can imagine, just as with a group of people, you put them together every day, they will behave slightly differently. Over the years, the behavior will change and so on. And that's exactly what happens. So you have a rewiring of the network of interactions. So today, if any of you form a group, that individual is the leader of the group, and you know that if you want to destroy function in the group, you remove that individual. But over time, when that group becomes a more discrete entity, more collective entity, suddenly that individual, removing that individual doesn't really affect the functioning of the group. And that's exactly again what happens in diseases. We're used to thinking the mutation in a gene causing, leading to a dysfunctional protein may initiate disease. But as the disease develops, that protein starts playing less and less important role, and suddenly the behavior becomes far more complex, far more dynamic, and far more non-linear. So even if you were to block that, you don't see much of an effect in terms of the therapeutic benefits that you were expecting. And that's is the major challenge today, and particularly for, at least for the prominent diseases that we deal with today. So trying to understand this suddenly calls for new disciplines, new approaches, and so on. So, so the biggest challenge we have is, how do I identify a target or a group of targets that I can hit in terms of drug molecules that I develop. And today, at least in my view, that is the biggest challenge that we face today. And the only way we can address that is using techniques, certainly we in India know all well, which is the techniques of Jugaad. You have to pull in out-of-the-box thinking and different strategies. And when you want to analyze complex behavior, you cannot escape mathematics. Understanding complex behavior, quantitative traits of complex behavior, etc., need mathematical principles. And since you're talking about large numbers of molecules at a given time, you need to integrate that mathematics with computational tools. So in a sense, you need now to take your high throughput biology work that you do, bring in mathematics, bring in computational science, integrate them all and somehow try to figure out what the targets are. And then of course that comes in with pipelines of chemistry and pharmacology downstream. So essentially this was the kind of principle that we founded the Drug Discovery Center on. And we work on metabolic disorders, which is diabetes, obesity, and cardiovascular disease. They're all linked together. So you may start out as a pre-diabetic, and then when you become a diabetic and as you progress, you start getting other complications, right? A heart or kidney or whatever. So this is again, so when you're talking about these diseases, you're talking about diseases that evolve over time. And that's what I mean. So you need to understand complex behavior of molecules, whether it's in the liver or the skeletal muscle or the adipose, and also understand how those interactions evolve to drive progression of the disease. So you now need to think not just of biological reactions or biochemical interactions, but how they evolve and to drive progression of disease. So suddenly the dimensions of time now come in when you want to think about targets. And these are challenging. So Dr. Madhura talked about this new center we founded, and it's basically trying to attain these kinds of goals. So we monitor disease progression using model systems, looking at how networks change in terms of proteins and metabolites, trying to see how these gangs evolve as time goes on, and can you identify which are the key guys in that gang who are responsible for spreading disease. And for that we use, again, like I said, Jugaad. To give you an example, you can use mathematical principles of how a rumor progresses in society, right? So if I tell somebody something and that somebody just transmits exactly what I've said, it will not spread. Beyond a couple of people, they'll get bored and drop out. So if somebody adds some mirch masala, then it actually catches life, spreads through a society as a rumor. So that's exactly what would happen in biological networks that are driving disease. So there would be certain proteins that amplify the signal. So you can use, as an example, you can use some of these rules of how networks, rumors spread in society, and try to see if they would apply to understanding biological networks. Who are the guys that amplify these signals? And if you can find that out, now if you block it, can you actually block progression of disease? So some of these, I give you another example, and that turned out to be fairly successful. So tuberculosis was mentioned, right? And we know that we have drugs against TB, but we also know that TB is now becoming resistant. And today you now have, certainly in some parts of India, strains of tuberculosis that are resistant to all drugs. And that's the problem, because we take this traditional approach of, if you want to kill a bug, develop an antibiotic that will kill it, right? So a few years ago we tried something different. So you know that TB infects a cell, your macrophage, and it stays there for extended periods of time, and that's really the site of infection. So the idea was, if I now develop a new antibiotic against TB, we know for certain it will come back with new strains of resistance. But at the same time, for TB to survive in that cell, it has to use the molecular machinery of the host cell, right? It has to co-opt it, it has to use it for its own ends. So it's infecting a macrophage, which is a very potent defense cell that you have, an immune cell that you have. So the first thing the microbug has to do is tweak its machinery to prevent it from unleashing its defense weapons, number one. And since it's going to, that's going to make a life inside the macrophage, it also needs to find mechanisms or tune mechanisms of the host cell by which it can access nutrients from the host cell, right? Particularly these are fat, lipids, and so on. So the idea was, instead of touching the bug, can you prevent it from manipulating the host macrophage, okay? So the idea, you needed to know which proteins it uses. So obviously the way it can manipulate the machinery of the macrophage is by controlling functions of certain proteins. So the first job was to identify those proteins, did that, and just to cut a long story short, that has now led to now what we have is a new drug for tuberculosis that does not touch the bug, prevents it from manipulating the host cell, and the host cell can now throw the bug out. And because it's not touching the bug, it doesn't care if the bug is drug sensitive or drug resistant or even totally drug resistant, right? So just switching perspectives a little can actually give you new. And this is now going close to phase 1 clinical trials in the next few months. So that's really what the Drug Discovery Center is all about, a bunch of crazy people trying to find out-of-the-box solutions by combining different approaches. But I think that's critical. We learn all these techniques and tools, but the care is not to, not to get, you shouldn't become automatons, and particularly in India, as again was mentioned, we actually have the problem of convergence of multiple diseases, right? We have diseases which are non-communicable, like diabetes, cancer. We also have this heavy load of infectious diseases. And the worst part, what we're realizing now, is they integrate with each other. As a diabetic, one is more susceptible to tuberculosis. As a diabetic, one becomes more susceptible to cancers. HIV infection gives you others, as you know. So we also need to think beyond simply one disease, one drug, but perhaps start thinking more in terms of how diseases converge to increase pathology, and how does one deal with that. And particularly in countries like India, that's a very, very critical question. So this has to be the core in terms of asking these questions, finding unusual solutions, And of course, all of this technology will then help you reach, but the idea is always preeminent. And the craziness is always the most demanding aspect of what you do. So good luck to all of you. I'm sure you'll enjoy this course and find it very, very instructive, and all the best. Thank you.
Thank you very much, Kanuri. You know, I just happened to visit Professor Kanuri's institute just 2 weeks back, and I was really impressed in a short time the kind of facility he has developed, which outreaches several of the international facilities I have seen across the world, where the drug discovery and actually the high throughput screening and all is done. I think he has done he has created an excellent facility. I think it is in a short time of a year or so. And may I throw a proposition? If you would have our trainees visit your center on one of these days, you know, if we can organize a bus for them and they can visit you, that'll be wonderful, because it will further add a value to where you want to do research, what kind of research in drug discovery you want to do in India. That will help you to actually see the facilities what he has created. So we will try to work out, no promises. He'll promise us a lunch for all our trainees if they go there. So, and we will like to visit your, all the trainees would love it. So now we come to our main guest, Professor Tekwani. I know everybody calls him Babu. I know him from my earlier days when I entered into research at JNU. He was at CDRI. And it just happened that we did our postdoc. He preceded me in Penn State University, where we both worked on polyamines. And then he moved on. I mean, this is my introduction of Tekwani, because that's how I know him. But we've been interacting at several meetings. He just organized a meeting at Spain where I was with Rafa at Madrid, and that was, previous one was at NATA, again on global infectious disease in Spain, and he's done some excellent work. Then he left off the shores of our country and he's moved on to, I'll tell you why. He moved on, and I think for better future maybe. So let me introduce Professor Tekwani, and I'm really grateful to you for taking this initiative. You know, I have to tell you, preparing all these lectures is quite a job, because it requires, because he has catered the course which will start you from drug development to how you do all the different models can be created, because those are very important part of this drug discovery research. And he's a pioneer in that field, and he has a very established lab in the US. He completed his PhD degree from Lucknow University, and he was a senior scientist at CDRI in the biochemistry division in Lucknow. He is currently a principal scientist at the National Center for Natural Product Research, and also a professor of the Department of Pharmacology, Pharmacy, University of Mississippi. He is a renowned specialist in natural products and drug discovery for malaria, trypanosoma, and leishmania parasites. His research interests include molecular basis of pathogenesis of disease caused by eukaryotic pathogens, in vitro drug metabolism, drug-drug interaction, development of high-throughput screening assays for drug screening. He is also the recipient of various prestigious awards and fellowships, one of the recent being faculty awards by School of Pharmacy, Cumberland Pharmaceuticals. I now request Professor Tekwani to say a few words about the course.
Maybe it's not going to be a few words, it's going to be a real kickstart, so I would like to put on the PowerPoint presentation.
I was thinking we'll have a tea break. Now you
It's
just
only 15
introduce that we'll do.
To please take the seats there. Sorry for the distinguished to make them sit a little longer. I just wanted everybody to have a feel of this course, so including trainees and all the chief guests and the distinguished guests also. So basically, I just wanted to do a kind of kickstart today and give you an overview of what you are going to learn in this course. Is going to be quite brief or maybe an important part. I really am sorry for the spelling mistakes here, biotechnology. When we submitted the original proposal, this spelling mistake was there and it was carried over all through this place, So as you know that this course is on bioefficacy and bioassay and bioefficacy model. Professor Kanuri Rao has already given you a good overview of overall process of drug discovery. This is a very small part of that drug discovery process. So what you are going to learn is that the knowledge which has already been generated through different approaches and how that knowledge is used for development of models, particularly for bioassays and bioefficacies. You will learn why I use these two words separately and why specifically I have capitalized the A in assays, because just to emphasize that these assays and efficacies are really important. So basically tools, targets and technologies, paradigm shift in new drug discovery and the biotechnology research. So we are going to have just kickstart today. Basically the objective of the course is going to be whosoever will be attending this course will gain the knowledge on principles and practice for discovery of new bioactive molecules. Bioactive molecules is very important term I am going to define in a short while, particularly drugs, biologicals and biotechnology products. Then you all will also acquire the critical knowledge about the current and recently developed tools and technologies for discovery of these bioactive molecules. Then bioactive molecules is actually I have tried to define in a very simplistic form is that a bioactive molecule has an effect on a living organism, tissue or a cell. Particularly in the field of nutrition, bioactive components and the compounds are distinguished from the essential nutrients. The nutrients are essential to the sustainability of the body, but the bioactive compounds are not. They are not essential since the body can function properly even without them, or because the nutrients are able to fulfill the same function. So don't confuse between the indigenous nutrient molecules and the bioactive molecules in that term. Then the bioactive compounds can have an influence on the health and functions. That is the important and the most important aspect why we want to work on the bioassays and bioefficacy models. Then bioactive compounds are found in both plants. Of course, originally the bioactive molecules, they were discovered from the natural product resources, particularly from the plants, animal products and several other natural product resources. But the important thing is that most of these bioactive molecules can be chemically synthesized, and that has been the important breakthrough in the sense that if we can synthesize these bioactive molecules naturally, all the time we need not to depend on the natural product resources, particularly for resourcing. Now, what will be your take home when you complete the course? I am trying to tempt everybody to stay till last, so I am just trying to set a stage here. So what will be your take home? The knowledge gained, whatever knowledge you will gain here during the course of this course will help you to develop the basic understanding of the subject. Particularly, you will have a more understanding. Most of you are already the students or the faculties or research or scientists in biotechnology, life science and biology. But once you complete this course and go through most of the modules, you will have a better understanding of the subject. Then you will be able to develop the multidisciplinary approach and strategy for product development, research and education. So I am going to touch many aspects of this area, so that will be one of the important take home. During the course of this lectures, we will try to do some case studies and problem-based learning exercises. This will provide opportunity to apply the knowledge for developing original research projects and also conceive some innovative ideas. So we'll try to do that during the course. The most important is that you will get an opportunity to apply the basic subject knowledge in life sciences, biotechnology, biochemistry, microbiology, immunology, molecular biology, whatever you are expert into, pharmaceutical sciences, pharmacology, or medicinal chemistry and biology, you will be able to apply this knowledge for the new drug discovery and biotechnology product development. You will understand that how that knowledge has been really used for development of bioassay and bioefficacy models. So that is an important aspect. Now, one of the things which Professor Kanuri Rao mentioned about the technological advancements is that biotechnology, life sciences, and modern drug discovery programs have been greatly benefited from a number of recent scientific and technological advancements. And what are the driving forces or the driving forces that? I will go one by one then. Rapid advancements in the bioinformatics, genomics, proteomics, and metabolomics, these have been really tremendous during last few years. Then availability of fully annotated genome databases. This has been the really breakthrough, particularly for the area of drug discovery and drug development. Several new tools have been developed, particularly structural elucidation, expression profiling, targeted gene disruption, in vitro cell technologies, cell biology and imaging technology. They have added another dimension to that. Discovery of large number of validated chemotherapeutic targets and the several new and unique metabolic pathways have been discovered through these annotated genome databases. And of course, the big data analysis, system biology networks, pathway analysis tools, these have further added to this knowledge and worked as a driving force. And of course, because now, particularly whenever you want to develop a product or drug, particularly for the human health, particularly for the human consumptions, not only for human consumption and even for the livestock and the animal health, the regulatory requirements are very stringent. So your initial bioassays and the tools to screen those molecules has to be very particular. So because of that, collectively there had been, these advances have fostered a kind of paradigm shift in the applied biotechnology and new drug discovery approach. Then the next one is when we talk about the bioassays, bioassay means which determines the biological activity of a molecule on a particular target or a cell. But then when we talk about bioefficacy model monitors the functional bioresponse of a molecule on an intact system or a cell or a whole cell animal. They have important application in discovery of these drugs, biological and biotechnological products, as well as understanding the molecular and cellular functions. So this is one of the reasons that we want to know more about these bioassays and bioefficacy models and how to really apply it for this particular kind of area. So discovery of bioactive molecules primarily depends on regulation on a variety of phenotypic, cell-based and pathway-specific molecular target-based assays. This is one of the requirements when you want to classify a molecule or a compound as a bioactive molecule. Then the cell culture-based phenotypic screening approaches, they have gained better acceptance because most of the phenotypic-based cell-based assay employ live cells. They have the capacity to predict in vivo pharmacological response in the early phase of discovery, particularly the bioactive natural products. Then the pathways and target-based screening technologies hold significant promise due to recent technological advancement and for understanding the molecular mechanism and action of bioactive molecules. So with this one, what I have planned for you all is divided this course into two modules. The module 1, we will cover the principles and technologies for bioassays and bioefficacy models. We will go through first the pathways and current approaches for discovery of new drugs and biotechnology products, because going to the real assays is really important for us to learn that what are really the pathways and principles for discovery of drugs and these biotechnological products. I will go through that. Then I will cover the phenotypic cell culture based assays, stem cells, 3D cell cultures, reporter proteins, and transgenic cell lines. How do we use this in bioassay models? I have included this particularly as a separate lecture because this is one of the important things where major advancement has been there in terms of screening bioefficacy and bioassays is the cellular imaging technologies, particularly high content analysis, high throughput screening. We will touch upon the instrumentation and the database analysis also, which is very complex process. Then the fourth lecture, I will try to go through the laboratory animals, transgenic animals. Specifically, I will tell more about the humanized animal models, which are really making breakthroughs in early preclinical studies of the drugs and biological products. Then the last one in this module will be regulatory compliances and good laboratory practices. Because this is very important for us to understand that in the current scenario, what are the regulatory compliances we have to follow? What kind of important processes are there when we are doing human studies, when we are doing animal studies, what kind of important aspects we have to learn. Otherwise, a single very little here and there can affect your future research. So after completing this module, we will go to, during this module, we will try to do some interactive case studies, particularly writing a protocol for IRB human study, writing a protocol for iCook animal study protocol so that we are able to learn how to submit this protocol for study, particularly for compliances. Once we complete that, then we will go to module 2, particularly application of these bioassays and bioefficacy models for particular therapeutic areas. It's very difficult for me to cover all the therapeutic areas, so I have included some of them where I personally work into and some of them which are important. I'm sorry if I'm not able to cover the area which is of your particular interest, but somehow during the course of that, something will come up. If any of you is specifically interested in something, please let me know. I just want this whole process to be very informal and very interactive. So since I work on natural products and we do a lot of studies on natural products, so I will cover in detail the bioassays and pharmacology of natural products in a separate module. Then bioassays and bioefficacy model for anti-infectives. We will try to cover the antibacterial, antifungal and antiviral drug discovery. Then also the bioassay and bioefficacy model for neglected tropical parasitic diseases where I personally work on. We will be covering malaria, leishmaniasis, human African trypanosomiasis and Chagas disease. These are the most important neglected tropical parasitic diseases. Why we want to do that in each lecture, I will give you the importance of that. Then we will also cover the models for metabolic disorders, particularly diabetes, obesity and cardiovascular antioxidant. We will try to cover in a whole process because most of these metabolic disorders are highly interrelated, and a lot of bioassays, they commonly cover all these therapeutic areas. Then finally, we will also touch upon the efficacy model for neurological disorders. I do some research in this area also. That's the reason I have particularly included this area, especially for neurodegenerative disease and nerve injuries. What kind of models we are using, what kind of models have to be used. So during the course of this module, particularly the application module, we will try to do a case study, particularly the discussion for developing and writing an original research proposal. So once I get to know each one of you, sometime tomorrow we will have an individual introduction and get to know that in which area, what you are working, what is your background. We will try to develop that kind of case study, particularly for developing and writing an original research proposal for new drug discovery and biotechnology product. So this is an overall overview we are going to cover. If you feel that this is going to be good, you know that beside you, any of your friend around probably did not know much about this course, might be interested, please bring him or her over. It will be good because the resources we are putting into, the efforts we are putting into are already there, and we would like as many people as to be benefited from this endeavor. In the last, I would like to thank the host faculty for taking the pains for organizing all these. Actually, the idea for this course was developed when I was working with Professor Madhubala on a project which we submitted to Indo-US Science and Technology Forum. That was a very huge project we submitted. Mostly the money was only for the travel. We were very hopeful, but we did not get that project. But during that interaction, she told me about this GIAN proposal, and then I told her about this idea, and she liked it very much. And through that, I got in touch with Rupesh Chaturvedi, who has worked with me when I was at CDRI, and then she brought in 2 more host faculty. Collectively, we have been able to put together a very impressive and very useful program for you all, and hopefully you will enjoy that. But beside that, I would like to thank Professor Patnaik, who is the GIAN coordinator, and also Professor Prasad, who was kind enough to give me a JNU fellowship and make me to able to stay at that guest house. So I would like to thank you all for staying over, and especially the distinguished for giving me the time for this. And I hope that you will all like this course in the coming course of days. Thank you.
Thank you very much, Professor Tekwani. Looking at your course, it looks like a very intense course, and I'm sure you all are going to gain a lot from this course. And it's not going to be easy, busy time, but he's a very good teacher, and he interacts well. And he, you know, as you have seen, it's a very, very intense course, and he has gone through every detail possible in drug development process. And he works on natural products, he works on drug discovery, and so it will be very helpful to all of you. May I now request Dr. Rohini Muthuswamy to give vote of thanks.
The onus is on me to give the vote of thanks. I would like to start with thanking, of course, MHRD for putting this course together, and their GIAN coordinators, both at the national level and at JNU. In particular, I'd like to thank Mr. Ram Singh, because he was at the GIAN office, because he was so helpful when all these glitches happened about the participant registration. I just had to call him and he would resolve the problem for me. So I really like to thank Mr. Ram Singh, our guest faculty, for agreeing and putting together this wonderful course. I would also like to thank the director, Jawaharlal Nehru Institute for Advanced Studies. We requested him for accommodation, and he was very cooperative, and that is where Professor Tekwani is staying. Then the next in line is the director for the Human Resource Development Center, and the staff at the center. Many of our participants are coming from outside and had requested accommodation, so we asked the HRDC whether they can provide us with some rooms, and they very graciously agreed, and so we have now 5 double rooms for our outside participants to stay. So that was very nice of them, and it resolved a lot of issues for us. Then the school, the dean, and the staff who helped us put this together. My co-organizers, Professor Madhubala, who was really the force behind this, Dr. Rupesh Chakravarti and Dr. Manoj Munde. And finally, I would like to thank the students and the staff in my lab and Professor Madhubala's lab, who helped us behind the scenes, things like the registration desk, getting the stuff ready. So they were the ones who helped us with, so I would like to thank them. And now we... Yeah.
Thank you, Rohini. I got to mention here my co-host here, Rupesh, could you identify yourself? Rohini and Manoj, please. Manoj is a chemistry person, so he would be, he'll be here for some of the lectures. You could interact with him. He also works on drug development. He's in School of Physical Sciences, and Rupesh is in Department of Biotechnology, and he's also heading the R&D in JNU right now at the university level. So I'm sure you'll be available here. And Rohini is a molecular biologist, does some excellent research, and you could get to talk to her also while you are here. and of course meet all the faculty of School of Life Sciences. Just few announcements. We have a tea break now, and after that I request all the trainees to come back, and we have an introductory session of each one of you, your background, so that the course faculty knows as to what is your background and how to manage his lectures so that it is understood by all of you. So he will be meeting after the tea break. We'll all come back here. And anything else? Tomorrow morning we meet at 9:30 here, and we'll put the schedule of all the lectures, and 9:30 till 12:30 we have a break in between. So there'll be 2 lectures per day. And Dr. Kanuri Rao has very kindly agreed to host all the trainees at the Drug Discovery Center at Faridabad. I'm taking the opportunity to make an announcement. The actual date will be announced once he goes back and lets us know that. And both the coordinators of GIAN program have kindly agreed to provide us with a bus to go to Faridabad. Thank you very much. And I look forward to all of you enjoying this course and learning from it. And we do have a evaluation form at the end of it where you can give your comments about the course, which will be very helpful for us to how we can improve the course if there are any flaws. Thank you very much.